Hi team, and welcome to this World of Medics video on stem cells. So let's dive right in and define exactly what we mean by a stem cell. A stem cell is just an undifferentiated or an unspecialized cell, which then goes on to further divide and produce daughter cells. And stem cells can either be called pluripotent stem cells, or, like many things in medicine, we've got a second name, which is haemocytoblasts. The best thing to do to illustrate what happens when a stem cell divides is just to draw a diagram. So we've got our stem cell here, and this is going to go on and produce two further cells through division. And the first of these cells, this is actually going to stay as a stem cell. And obviously this is going to go on and then further divide and go through this process again and again multiple times. And the second of these cells, this is go on to become a differentiated cell. And this obviously depends on what tissue this is going to be in and what area of the body this is in. And when this occurs within blood and forming blood cells, this is called haemopoesis. Or alternatively, it can be called hematopoeisis. So you might hear both terms being used. So the process of stem cell division, this can be the rate at which this happens. This can be affected by various things. And the first of these is actually the location in the body where the stem cell is. So if, for example, the stem cell was in the skin, this is an area of a high rate of turnover, therefore meaning that stem cells will divide at a relatively high rate, and this could be as much as once a day, for example, especially in people who, for example, the skins of their hand, the palms of their hands, undergo a lot of abrasion day to day. So gardeners, climbers, weightlifters, for example. An area of the body where we might have a relatively low turnover of cells and therefore a low rate of stem cell division will be connective tissue in adults. Another thing which can impact the rate of stem cell division is a person's age. So if we have children or newborns, they're going to generally have a high rate of stem cell division. And obviously this is because a child is still growing and therefore they need to produce more cells, but also because they have a higher amount of red bone marrow, which is where stem cells tend to divide, and especially where haemopoesis happens, so blood cell formation. And conversely, in adults, in comparison, the rate is going to be lower. And another thing which is worth mentioning is the amount of ATP available within the body. So ATP, as we know, is our energy source, which is utilized by the cell to undergo various processes, including stem cell division. Therefore, if we have a low amount of energy or low amount of ATP within the body, stem cell division can therefore be halted or slowed down. And well, when might this happen? So if a person's starving, this is a characteristic time where stem cell division might be halted or be impaired. And this is obviously going to affect childhood growth, wound healing, things like resistance to disease, and also can change the lining of the digestive tract. So it's important to be aware of this. Now, we touched on red bone marrow, so let's go ahead and look at that in more detail. So red bone marrow. Now, as you've probably guessed, this is found in bone. So let's draw a very rough diagram of a bone, and you'll have to use your imagination for this. So the red bone marrow is found in the spongy bit of bone, and within the spongy bone we've got this structure which essentially is like a mesh, and this is called trabeculae, and it's actually within the spaces found amongst these trabeculae that we get trabecula, or trabeculae for plural. And these red bone marrows, and this area of red bone marrow is very highly vascularized, so it has a very dense blood supply. And one of the reasons for this is that the cells produced can easily go into the bloodstream through structures called sinusoids, which are just capillary membranes essentially. and these will eventually drain into blood vessels. So within the red bone marrow itself, there's only about 0.1% of the cells which are going to be stem cells. And the red bone marrow, as we said, it's more likely to be found, or there's a higher amount of red bone marrow in children. But one of the reasons for this is that the red bone marrow, as you get older, it gets converted into yellow bone marrow. And the yellow bone marrow has mostly fat cells. So we'll say with age, this gets converted into yellow bone marrow. However, in severe circumstances, such as extreme levels of bleeding within the body, the yellow bone marrow can essentially become red bone marrow again as stem cells infiltrate it and then start to divide within the yellow bone marrow. But that's in extreme situations. So we know about haemopoesis now and this basic concept of stem cells dividing, but let's take a closer look at the different pathways that take place when a stem cell divides. So this diagram shows exactly what we mean by stem cell division and what happens at each stage. So let's start from the start. So we've got pluripotent stem cells, or hemocytoblasts, at the start of this diagram, and they are going to go on and divide into two separate types of stem cells. And the first of these is your myeloid stem cell, and the second is your lymphoid stem cells. Okay. The first one that we're going to concentrate on is the myeloid stem cell lineage. So the next stage we've got, and we'll just highlight here by putting a star beside these terms. So this stage is actually your progenitor cell stage. And we've got terms here such as CFU-E. And the CFU part actually just means colony forming units. And the E here, in this case, means erythrocytes. And then we've got CFU-MEG, meaning megakaryocytes, and CFU-GM, meaning granulocytes and monocytes. So this is our progenitor cell stage. Now, when we move on from this, we've got our next stage, and we'll put little asterisks beside these terms again and put a little quote at the top. So these are your blast cells, or your precursor cells. And as we can see, they've got blast in the name, so that's pretty self-explanatory. So let's take a look at our first pathway, if you like, which is forming our red blood cells. So the pre-erythroblasts become reticulocytes, and remember, these cells lose their nucleus and then become erythrocytes, or red blood cells. And then we've got our megakaryocytes, which are going on to become megakaryocytes, and then our platelets. And then we've got myeloblasts, and it's important to note that this step skips the progenitor cell stage and goes straight to the blast stage to become a myeloblast. And then this goes on to directly form our three white blood cells: eosinophils, basophils, and neutrophils. Now, let's just touch on these three cells here. They can actually be classified as our granular leukocytes, meaning the granules within the cells are visible under the light microscope. And our last step here, we've got our monoblasts, which are then becoming our monocytes, and then they are going to enter the tissues and then become more differentiated and become macrophages. And these are the cells which undergo the process of phagocytosis, and where they take in foreign organisms and ingest them. So that's our myeloid stem cell stage. Next, we've got our lymphoid stem cells. So let's take a closer look at that. And again, this step, or this lineage, is actually just going to skip the progenitor cell stage and become blast cells. So we've got our B lymphoblast first of all, which is going to become our B cell, which is then going to differentiate into our plasma cell, which is involved in obviously producing antibodies. And then we've got our T lymphoblast, which becomes a T cell, and our NK lymphoblast becoming a natural killer cell. So these cells in our lymphoid stem cell stage, they are classified, as well as the monocyte, as being agranular leukocytes. And that's not because they don't have granules. They in fact do have granules, but the granules aren't visible under the light microscope. And there's a few things, so this whole process, there's a few things which actually stimulates different aspects of this process. So let's take a closer look at that. So the first thing that we're going to look at is something which stimulates the side producing red blood cells, and that's something called EPO, erythropoietin. And you might have heard of that. It's a substance which is produced in the kidneys which stimulates the production of red blood cells, and it affects these two stages which are outlined here. And we've got something else which sounds similar to EPO called TPO, thrombopoietin, which stimulates the production of platelets by stimulating the progenitor cells in this stage. And we've also got another substance called cytokines, and these are involved in the immune response in various ways, but in this circumstance they are going to go on and stimulate the proliferation of progenitor cells and blast cells. And there's two main types of cytokines that we want to know about. The first of these is interleukins, and the second is colony stimulating factors, so CSF for short. And these are involved in this process of hematopoiesis. So specifically, we've got our multi-CSF or MCSF, which is acting on our myeloid stem cells, and we've got GM-CSF, which is acting on our progenitor cells, which are going to produce our monocytes. And then we've also got our GCSF, so granulocyte colony stimulating factor, acting on our myeloblasts. And lastly, we've got our MCSF acting on our monoblasts and stimulating them to become monocytes. So there you go, that's a quick overview on stem cells, hematopoiesis and blood cell formation.